Hello everyone, I'm Ziwei Pan, a graduate student at Shengli's lab at the JAX lab. Today, I would like to talk about our work named DNA Methylation Calling Tools for Oxford Nanopore Sequencing, a survey and human epigenome-wide evaluation. 5-cytosine modification, 5MC for short, is a major DNA modification and can regulate gene expression. Traditionally, DNA methylation is measured by bisulfite sequencing, but the chemical conversion is harmful to DNA and is limited to short-range detection. Recently, third-generation sequencing, especially Oxford Nanopore sequencing, can achieve extra long-read sequencing at single-base resolution. Nanopore sequencing can monitor electronic current changes of DNA translocation at real time when nucleic acids pass through the protein nanopore. Since modified bases have signal shifts compared to unmodified bases, we are able to use nanopore sequencing to detect DNA modification. But extra methylation calling tools are needed to interpret methylation calling information. Currently, there are multiple methylation calling tools for nanopore sequencing. For model-based methods, such as nanopolish, deep signal, and McLendon, they used pre-trained model, while other methods, such as Tumble, rely on statistical tests for methylation prediction. However, there are no benchmark on 5MC detection from nanopore sequencing on human epigenome-wide analysis. Therefore, we would like to provide a systematic evaluation of methylation calling tools on human 5MC detection from nanopore rates. We prepared 4 datasets from nature human DNA and generated nanopore sequencing and bisulfite sequencing at the same time. We compared 7 methylation calling tools: nanopolish, McLendon, deep signal, Gumpy, Tumble, deep mode, Meteori. After base calling step for nucleotide sequence and genome alignment, we follow each tool's instruction to do methylation calling. Specifically, we designed a performance evaluation at single-molecular, single-base resolution. First, we evaluated the per-read 5MC accuracy on fully methylated or fully unmethylated sites determined by bisulfite sequencing. We developed 2 classification systems for evaluation: singletons, non-singletons, and biologically relevant genomics contents. Second, we compared per-site 5MC prediction by methylation correlation coefficient calculation and methylation pattern evaluation. Last, we did running speed and resource usage evaluation. The dataset quality summary after base calling step showed that all 4 datasets achieved comparable read length and high base quality. We first compared per-read methylation calling performance at singleton and non-singleton. The F1 score across 4 datasets showed that nanopolish, McLendon, and deep signal outperformed other tools, and all tools had lower accuracy at discordant non-singletons. We also checked the per-read performance on multiple biological relevant genomics regions. We found, again, nanopolish, McLendon, and deep signal achieved higher F1 score in genomics regions, but they are not that accurate in intergenetic regions. For per-site performance, the Pearson's correlation coefficient between predicted DNA methylation percentages and corresponding bisulfite sequencing showed that the methylation levels predicted by nanopolish, McLendon, and deep signal achieved highest correlation with BS sequencing, and they share similar binary methylation distribution. The relationship between CpG methylation and distance to TSS or CDCF binding site indicated that nanopolish sequencing can closely resemble the DNA methylation patterns from BS sequencing. Surprisingly, nanopolish displays the lowest DNA methylation levels at TSS. When comparing size numbers, we found that McLendon and deep signal covered more CpG sites than nanopolish, and the difference increased greatly for lower sequencing deep data size. For running time and peak memory, nanopolish and McLendon usually ran shorter than DeepSignal. Nanopolish consumed limited peak memory, but McLendon required a much larger peak memory for running, and there is a trade-off between methylation calling performance and running resources. To summary, the performance of methylation calling tools varied in diverse genomics contexts, CpG size coverages, and running resources at both single-read and single-base resolution. For initial DNA methylation calling, we would like to recommend nanopolish since it can achieve high performance with a relatively short running time and low memory requirement. For a systematic analysis, especially for low coverage data size, it will be more accurate to integrate nanopolish with McLendon or DeepSignal if more computational resources is available. Also, the bad performance in discordant non-singletons and intergenetic regions showed potential improvement for tool developers. We would like to thank the people listed here for helpful discussion on this work. Thank you so much for your time and attention.